Inside the nucleus of a human neuron, genetic instructions are packed with immense physical density. To find the highest concentration of inherited risk for late-onset Alzheimer's disease, we have to zoom in on one specific structure. This linear map represents the 13.32 band on the long arm of chromosome 19. It is the most heavily scrutinized address in the human genome. At the center of this band sits ApoE. For decades, the E4 variant of this gene has remained the primary predictor of genetic risk for the disease. But ApoE has an immediate neighbor. The TOM40 gene sits head to tail with it, separated by a gap of barely 2,000 base pairs. This proximity leads to linkage disequilibrium. Because they sit so close together, these two genes are physically tethered during inheritance, passing from parent to child as a single locked block far more often than random chance allows. In European populations, this phasing is rigid. The long variant of TOM40 is almost universally paired with the high-risk E4 variant of ApoE, traveling together across generations. This creates a methodological crisis. When two variables always appear together, standard statistics struggle to untangle which one drives the clinical risk. Statisticians attempt to resolve this through conditional regression. When the influence of ApoE4 is mathematically removed from the data, the remaining risk signal for TOM40 disappears. Because the signal vanished in the math, the field broadly dismissed TOM40 as a statistical passenger, a meaningless marker merely hitching a ride with its neighbor. Dismissing a gene based on its statistical shadow ignores the physical reality of the channel it builds. Understanding the disease requires separating the statistical ability to predict a binary risk from the mechanistic ability of a protein to cause cellular collapse. If TOM40 is merely a passenger, researchers must explain why targeting its specific biological pathway can rescue neurons from bioenergetic failure in preclinical models. The inherited TOM40 sequence defines the chronological timeline of the metabolic breakdown, shifting the investigation from whether the disease happens to how fast it advances. To understand the speed of that decline, we must leave the one-dimensional genomic code and examine the three-dimensional molecular machine constructed by the TOM40 gene. Alzheimer's is a physical disease of cellular architecture. In the neuron, the TOM40 locus encodes the most vulnerable load-bearing choke point in the organelle. The mitochondrion is defined by its physical boundaries, a double membrane structure that presents a nearly impermeable barrier to the rest of the cell. Having transferred nearly all its ancestral genome to the host nucleus, the mitochondrion must import every one of its working parts from the surrounding cytoplasm. The solution is the outer membrane translocase. At its center sits TOM40, a single beta-barrel channel. A constant stream of over 1,000 preproteins must thread through this door. This essential cargo relies completely on this singular entry point. This creates a physical bottleneck. The absolute capacity for mitochondrial resupply is limited by the number of functional TOM40 channels on the membrane surface. Because this channel controls the entire supply line, TOM40's patency determines the bioenergetic ceiling of the neuron. The import gate functions as the primary convergent lesion targeted by specific protein aggregates. First is a transit and inhibit assault. Amyloid beta peptides thread through TOM40 to reach the matrix. Once inside, they bind the enzyme ABAD. This disrupts the active site, generating reactive oxygen species and depressing complex 4 output. The second mechanism is a structural blockade, initiated when non-glycosylated amyloid precursor protein, or APP, arrives at the gate. The TOM receptors falsely recognize a cryptic targeting signal on the APP molecule and begin drawing it into the pore. However, an acidic domain within the APP sequence acts as a stop transfer signal, physically preventing the 770 amino acid molecule from passing through the narrow channel. The result is a transmembrane arrest. The protein wedges into the beta barrel, causing the import machinery to seize and permanently blocking the pore. This physical plug starves the matrix. Replacement respiratory subunits can no longer enter, leaving the mitochondrion unable to repair oxidative damage. This blockade triggers a third mechanism, the miscalibration of the PINK1 surveillance system. Normally, PINK1 continuously enters the TOM40 pore, passing to the matrix where it is degraded. But when plugged, PINK1 cannot enter. It accumulates on the outer surface instead, acting as a beacon that flags the mitochondrion for destruction. The APP plug tricks the cell into executing its own energy factories, even if those mitochondria are otherwise electrically functional. This physical fragility connects directly back to the inherited poly-T length polymorphism within the TOM40 gene. If poly-T length acts as a volume dial for TOM40 expression, it sets the baseline abundance of import channels established at birth. To isolate this effect, researchers exclusively studied patients with the E3-E3 genotype, where the ApoE-E4 influence is absent. Within this cohort, the long poly-T variant independently predicts a significantly earlier age of disease onset than shorter variants. In cognitively normal adults lacking the E4 allele, these long poly-T variants correlate with reduced gray matter volume in cortical regions affected earliest by the disease. These neuroimaging metrics quantify the trajectory and speed of preclinical metabolic decline. Once the statistical noise of ApoE is removed, TOM40 emerges as the genetic timer determining when a neuron's bioenergetic reserve will be exhausted. A 2024 discovery suggests the attempt to partition signal between TOM40 and ApoE may have been flawed from the start. Researchers identified transcriptional read-through, where the cellular machinery ignores the genetic stop signals and bridges the gap between the two neighbors. This process results in the T9A2 chimera, a hybrid mRNA molecule that contains instructions for both proteins. This creates a single biological machine, a TOM40 channel physically and permanently tethered to an ApoE molecule at the mitochondrial surface. At the level of the organelle, the channel and the lipoprotein function together. The statistical struggle to assign signal to one gene or the other was likely an illusion. This intracellular pathway exposes the limitations of current strategies, focused solely on clearing extracellular amyloid plaques. Clearing this extracellular debris has absolutely no effect on the specific proteins currently jammed inside the mitochondrial pore. An effective therapeutic strategy must augment the cell's own import stress clearance machinery to actively remove the physical plug from the channel. Sustaining neuronal function requires resolving the physical obstruction at the mitochondrial gate.